Hello, my name is Sequoia Burleson Ross, and today's presentation is on glycolysis, glyconeogenesis, as well as the enzyme Aldolase and the gene expression of such enzyme. Now, first, let me just say I am definitely a visual and hands-on learner. So I had to diagram out the processes myself in order to effectively be able to explain it today. So first, let's start off with the monosaccharides. So all sugars have to be based off of the simplest component, which would be the monosaccharides. There are three examples here. We see fructose, glucose, and galactose. Fructose is a ketone. What is a ketone? A ketone is when the carbon to double bonded oxygen occurs in well, not middle of the chain, but within the chain. While glucose and galactose are aldehydes where the carbon double bonded oxygen is at the end of the chain. Aldehyde. See? So now fructose and glucose compared to galactose, the difference is the arrangement of the atoms. So our hydroxyl group is on the opposite side. To represent these chains in their ring formation is down here, each one. These rings can bond and connect to make more chains of polysaccharides. The bonds are very dependent on, well, the breakdown of the sugars is very dependent on the bonds. There can either be alpha bonds or beta bonds. Beta bonds are not easily, well, not at all digestible by humans. We lack the enzyme to be able to break beta bonds. So therefore, sugars, complex carbohydrates that contain beta bonds are usually known as our dietary fibers. While we have the enzyme amylase, which is the enzyme that breaks the alpha-1,4 glycosid bonds that connect these rings. Now let's talk about glycolysis, and this is my big overview of glycolysis. Okay, break it down to smaller steps. Out of all 10 steps involve oxidation, but let's start off with what is glycolysis? Glycolysis is the breakdown of sugar. So we have glucose right here, our 6-carbon skeleton, right, denoted by the 6 black circles. For our glucose, the glucose enters the cell through a glut receptor. There are 4 different types of glut receptors, each specific in their location and their job. But glut receptor 4 is the only one that is insulin dependent, while the other 3 are independent of insulin. GLUT4 receptors are only found in muscles and adipose tissue, though. While all 4 types of glut receptors are indeed bidirectional, only glucose in its natural state can go either way through the glut receptor. Here I have the reaction written in red because once we have the enzyme hexokinase, specifically hexokinase 4, or if we're in the liver, then it's going to be glucokinase. This enzyme reacts with glucose, and then using an ATP, we lose a phosphate and make ADP, and now we end up with glucose 6-phosphate. So by tagging the glucose with this phosphate on the 6 carbon prevents it from leaving the glut receptor and leaving the cell. The next step from glucose 6-phosphate would be to transform into fructose 6-phosphate. Well, wait, glucose to fructose. Well, glucose, remember, is an aldehyde where the double bonded oxygen is at the end of the chain, and fructose is a ketone where it's within the chain. So we move our double bonded oxygen thanks to the enzyme phosphohexose isomerase. This enzyme and this reaction is reversible, which is why it's written in blue. Now, our second irreversible reaction is from fructose 6-phosphate into fructose 1,6-bisphosphate from the help of phosphofructose kinase 1, PFK1, and the phosphate from another ATP tagging the first carbon on the chain with another phosphate group. Bisphosphate because the phosphates are not located next to each other. They have at least one carbon in between, this case 4. Now, the enzyme aldolase is used to split the 6-carbon fructose. This fructose 1,6-bisphosphate is split by aldolase into either dihyd dihydroxyacetone phosphate (DHAP) or into glycerolaldehyde 3-phosphate (GA3P). GA3P and DHAP are can, thanks to the enzyme triose phosphate isomerase, these can be exchangeable. The reaction is reversible, so in the sake of glycolysis, we would want the DHAP to be transformed into GA3P. So theoretically, if we have two GA3Ps, then we'd continue along our pathway in this direction. So let's just focus on that. So from GA3P, we use our GA3PDH, the hydrogenase. Okay. In the reversible reaction, we use NADH or NAD plus to create NADH, taking a hydride. What is a hydride again? A hydride is a hydrogen proton with 2 electrons. So essentially a negative ion of hydrogen. So the GA3PDH adds a phosphate while we lose a hydride to make room for that phosphate. Now we have a phosphate on the 1 and 3 carbon, so we have 1,3-bisphosphoglycerate. Now with the enzyme phosphoglycerate kinase and an ADP now creating ATP, taking that phosphate group back, which before we had used up some ATP, so now we are creating ATP. We now have 3-phosphoglycerate. Now with the enzyme phosphoglycerate mutase, we can mutate this molecule and move that phosphate group from the 3rd carbon to the 2nd carbon. So now we have 2-phosphoglycerate. Here's the molecular structure. We see the double bonded oxygen here, and carbon here from our ketone. Now with the enzyme enolase, we move that double bonded oxygen, we move one of those bonds to the carbons. So now we have a double bonded carbon and a single bond to the oxygen. This now creates the molecule phosphoenol pyruvate, also known as PEP. PEP, then now this is our 3rd irreversible reaction. PEP gets turned into pyruvate with the enzyme pyruvate kinase and another ADP creating an ATP, taking that phosphate group back. Normally this is where glycolysis would end, but pyruvate can either take the aerobic pathway and create acetyl-CoA, or if there's no oxygen, takes the anaerobic pathway to making lactic acid, where the enzyme lactate dehydrogenase is used and NADH loses its electrons to NAD plus, where it's oxidized and the lactic acid has been reduced. Lactic acid can then travel to the liver, can be used to create glucose, ATP, but too much can lower the pH of the blood, creating it to be acidic. Okay. So now here's our big overview again. Hard to see, but with our glycolysis, we had used up 2 ATP, but if we had 2 of our GA3Ps, so we go through this cycle twice. We use ATP once, use it twice, now we make one again, now we make another one again, so then we make 4. So -2 plus 4, net gain of 2. And then we also gained 2 NADH molecules right here. NADH was made, and over with the, well, no, not there, but that happens twice. Okay, now this is a whole big overview of neogenesis on top of everything. So let's go into detail by starting at the end. Oh wait, where'd it go? There we go. Okay. So I switched acetyl-CoA and lactic acid for the sake of space. So remember, lactic acid was our anaerobic pathway where we used the NADH and left the NAD+, but now that is reversible with the same enzyme. So lactic acid can be turned into pyruvate. But what happens with pyruvate? Well, pyruvate has to enter the mitochondria. Once pyruvate is within the mitochondria, normally you would take the aerobic pathway and create acetyl-CoA and enter the Krebs cycle. This 8-step cycle can occur and will occur naturally anyway. However, once pyruvate is within the mitochondria, if we add some carbon dioxide and use the energy from an ATP and have the enzyme pyruvate carboxylase, we can make we can create the molecule oxalate oxalate acetate. OAA. No matter how many times I try to say that word, I can never say it right. So now with our OAA molecule, which is represented in its molecular form here real quick with our 4-carbon chain. We went from a 3-carbon plus our carbon dioxide to create a 4-carbon chain, and we have 3 double bonded oxygens and 2 regular. But OAA cannot leave the mitochondria on its own. It has to be transferred back into malate, which luckily is a reversible reaction that already occurs in the Krebs cycle. Malate is able to leave the mitochondria and it gets turned back into OAA. OAA, with the help of the enzyme phosphoenol pyruvate carboxykinase, PEPS-CK, we lose our carbon dioxide and some energy in GTP, and now we're back at our PEP molecule. So even if we start with lactic acid, which is stored in the skeletal muscles, lactic acid, now, reversing NAD plus back in NADH, but we go back to pyruvate, into OAA, into malate, back into OAA, into PEP. Now that we're at PEP and out of the mitochondria, we go from PEP, reversible reaction, reversible, reversible, and reversible. Now we're at GA3P. Once we get back to GA3P, we need to be able to get back to glucose, but we have to make sure we have both GA3P and DHAP. We need both of those in order to create fructose 1,6 bisphosphate. Luckily, GA3P and DHAP are interchangeable due to our wonderful enzyme here. So once we have both molecules, we're able to use Aldolase, our enzyme, circled now in red, to combine the 2 molecules into one for fructose 1,6 bisphosphate. Now this was our other irreversible reaction. Luckily, with a new enzyme, fructose 1,6 bisphosphase, we can go back to fructose 6-phosphate. This is a reversible reaction back to glucose. We went from our ketone back to an aldehyde. Our glucose 6-phosphate still has our phosphate group, so it can't go through the GLUT receptor directly. It now has to enter the smooth ER, where it comes into contact with the enzyme glucose 6-phosphase, and it gets transformed back into regular glucose, removing that phosphate group. And now it can freely enter and exit out of the GLUT receptor. But wait, there's more. Now we have glycerol over here, which is the head of a triglyceride. Once the triglyceride goes through lipolysis and is separated from its fatty acids, the 3 fatty acids go off on their own, and the glycerol is what's important in our reaction right now. So with glycerol and the enzyme glycerol kinase, we can create glycerol 3-phosphate, where we add a phosphate group to the 3rd carbon. The chain can unravel and become DHAP. Now, again, being interchangeable with GA3P, or if we are able to reverse our steps over on this side and balance out, either way, with both molecules and the help of Aldolase, we can now go back to our fructose 1,6 bisphosphate, and back, and now create glucose and leave the cell again. Now, Aldolase, the enzyme I had circled in red that was used to split and to recombine our 2 molecules, back here, GA3P and DHAP. There are actually 3 classes of this Aldolase enzyme. Ugh, too far. Okay, Aldolase classes 1, 1A, and 2. 1 and 1A are very similar. They go through the same mechanisms, but they actually have different structures. Aldolase class 1 is the most common. It occurs in a lot more organisms, and it's typically formed into homotetramers. So there's 4, and all 3 of these classes contain TPI beta alpha barrel folds. So 1, 2, and 3. But now class 1A only occurs in archaea, but that's because it also it's larger, octamers, decamers, and hyogomers, so 8, 10, and more. While class 2 of aldolase are usually found in other eukaryotes and smaller organisms. It has a much simpler mechanism of action, which all these mechanisms will be demonstrated in a moment. Plus, these are homodimers, so it's just 2 mirror images of each other. All right, so class 1, the ones that we have of aldolase. This is our DHAP, this is our aldolase, and down here we have our GA3P. So DHAP is attracted to our enzyme. Our enzyme helps to encourage the transfer, well, base is included. This is our shift base formation, right? So the base transfers its electrons, and then there's a chain. We follow the arrows for the electrons. Boom, we're able to bond DHAP with GA3P. Now we have the bond, and then we return the electrons back to the base, and the enzymes are able to release our now fructose 1,6 bisphosphate. Another way to understand this reaction is to watch this amazingly demonstrated and detailed video where it is walked through one step by step. The key reactions of aldolase, the formations of such, and the combinations, and with the overall reaction, and each step involving the enzyme's active site. Yes. And how the electrons are transferred. So class 2, which is not as common, especially not at all happening in humans, but is still good to look at because it's much simpler. The aldolase enzyme is able to just donate its electrons to DHAP, which is then able to bond with GA3P, and then it bonds and it gives the electrons back to our enzyme to create our fructose 1,6 bisphosphate. Now, aldolase, especially class 1 aldolase, has 3 isoforms. There's aldolase A, B, and C, respectively. Aldolase A is found in muscles and red blood cells. Its genes are located on chromosome 16 in humans. Aldolase B is located in the liver, kidney, and I think I said river. I meant liver, kidneys, and small intestines. The genes are located on chromosome 9. And aldolase C is usually found in the brain, smooth muscle, and nervous tissues. Its genes are located on chromosome 17. To represent the fact that not only are all 3 of these enzymes needed for both glycolysis and glyconeogenesis, but the slight variations between A, B, and C. These are all different aldolases, and the different colors represent the different variations. So again, these are located in different parts of the body, right? But we see here our blue, which is A, and our B is green, and red is C. These ball and stick structures represent the active sites of each of these enzymes. Now, for the gene regulation of aldolase B, there are 3 major regulators involved. The first is HNF, hepatocyte nuclear factor. There's technically 4 of them, actually, but importantly, 1 would be a strong activator, while 3 would be a strong repressor. And then 2 is actually non-competitively inhibits DBP. What is DBP? Well, DBP is the promoter protein that binds the D side of albumin, and it's a strong transactivator. So is not strong. CEBP, which is CCAAT, enhancer binding protein. Both of these are highly specific in hepatic tissues. As a flow of diagram to represent the regulation and when each of these molecules comes into play, maybe not each, but there is the chain of events and series of reactions and consequences of such based off of all of these regulators and how it can affect aldol, aldolase B. Here are my references, and I would like to thank you for taking the time to listen and watch my video. And if you have any questions or comments, please leave them below. Thank you.